respond to wind orientation preference. When we activated these neurons, we're activating them all at once, not in a particular column, but we saw that we were able to produce these sort of directed, sort of curling, searching trajectories going off in a particular direction, which was quite interesting to us. And when we imaged from them, we saw responses of sort of two flavors. The first was that we got column-specific direction-selective odor responses. So here I've taken an ROI around just one column in the fan-shaped body. And what we're seeing is that they seem to have the strongest response to one particular direction. We saw another type of response as well, which is where the flies seem to respond just to one particular direction and not the others. So what this says is that it appears as though these neurons are capable of integrating this wind direction and odor information and responding in a specific way, only to one direction, unlike what we've seen before. So in summary, today I told you about how we designed an optogenetic activation paradigm to identify the neural circuits involved in olfactory navigation. I showed you that higher order olfactory neurons promote both upwind runs and offset search, and that these neurons respond non-directionally to odor. We then traced this pathway deeper into the fly's brain, into the fan-shaped body, part of the central complex. And here I showed that there's pure odor information as well as pure wind direction information going into the fan-shaped body, and that a particular subset of local fan-shaped body neurons are posed to integrate this information, picking up both wind direction and odor information, to turn that into a directional signal for the fly. So thank you for listening. I'd like to thank a couple people. First, Kathy Nagel, my mentor, the people who worked with this on me, Aaron Lands, Tim Courier, and Angela Licata, as well as the rest of the Nagel lab, the Chopik lab, who we share space with, as well as the Staropoulos lab and the Syed lab at University of New Mexico, and then my thesis committee, Dima, Niels, and Nick. And I can now take any questions.
Awesome, thank you very much for this great talk. Amazing data. And we are ready for questions. Panelists as well as attendees can ask any questions. Don't be shy. Okay, then I just wonder, like, what do you think would be the next step for you then? Because it seems like pretty much lined up everything. So what are the non-answered questions there?
So I think one of the things we want to look at next is, these responses are done in a fly that's basically hanging under a 2-photon microscope in the dark. So they have really no context of how the world around them is. So one of the things we want to do is put the fly on a walking ball and be able to have them like see their real environment and then activate, present the same stimuli and see how the flies respond to that. We're also thinking about sort of wanting to activate individual columns at once. So here we're activating all the columns together, which is a bit confusing to the fly, I'm sure. That's not how it would naturally be based on the physiology. So we're hoping to use some genetic tricks to label either a small subset of columns or only single columns, activate and see how the flies respond.
Okay, great. Great, thank you. And I think we can start moving to our next.